To start with, what is the coronavirus anyways? COVID-19 is part of a family of diseases called Coronavirinae. The virus causes diseases in mammals and birds. SARS, that was also identified in an epidemic in 2003, is also a disease caused by coronavirus. Symptoms of COVID-19 include fever, dry cough, tiredness, diarrhea, and many more. So what caused the initial spread of the virus? Based on initial genetic testing in past outbreaks, it is likely that the coronavirus first originated in bats. This is because the closest relative to the Wuhan virus was a virus from bats, which was 96% similar. Even though the theory that the virus originated from bats is relatively accepted between scientists, how they spread from bats either directly or through some other organisms is still unknown. There is a theory that the virus in bats was contracted by dromedary camels and into the human population. However, there are also theories by Chinese scientists that pangolins may be the middleman. Though bats and camels and pangolins may be the source of the virus, they are not the villain. If anything, the humans are the problem as we are enclosing on their domains. The structure and characteristics of the coronavirus. Because most viruses are extremely well adapted to their host organism, virus structure varies greatly. However, there are some general structural characteristics that all viruses share. All viruses have a capsid, or head region, that contains its genetic material. The capsid is made of proteins and glycoproteins. Capsid construction varies among viruses, with most being specialized for a particular virus's host organism. Some viruses, like the coronavirus, have a membranous envelope surrounding their capsid. This allows viruses to penetrate host cells through membrane fusion. The virus's genetical material rests inside the capsid can be either DNA or RNA, or even in some cases a limited number of enzymes. In addition to the head region, some viruses, mostly those that infect bacteria, have a tail region. The tail is an often elaborate protein structure. It aids in binding the surface of the host cell and in the introduction of virus genetic material to the host cell. Coronaviruses are large, roughly spherical particles with bulbous surface projections. The average diameter of the virus particle is around 125 nanometer. When the spikes are viewed under the microscope, it is viewed like a crown, which is why the name contains the word corona, which is Latin for crown. Coronaviruses contain a positive sense, single-stranded RNA genome. The viral envelope consists of a lipid bilayer, in which the membrane, envelope, and spike structural proteins are anchored. Inside the envelope, there is a nucleocapsid, which is formed from multiple copies of the nucleocapsid protein, and are bound to the positive sense single-stranded RNA genome in a continuous beads-on-a-string type conformation. The lipid bilayer envelope, membrane proteins, and nucleocapsid protect the virus when it is outside the host cell. How does the coronavirus infect humans? Infection begins when the viral spike protein attaches to its complementary host cell receptor. After attachment, a protease of the host cell cleaves and activates the receptor-attached spike protein. The cleavage and activation allows the virus to enter the host cell through one of the two methods, endocytosis or direct fusion of the viral envelope with the host membrane. The first method is direct cell entry, where the protease of the host cell cleaves and activates the protein, and triggers the process of membrane fusion, before injecting the viral genome to the host cell. The other method is endocytosis. This is a process by which material cell, after being surrounded by an area of the cell membrane, which then is pulled into the cell, forming a vesicle. Once inside the cell, uncoating occurs where all the protein are removed, leaving the contents of the virus itself, which is the genomic RNA, and will be released into the cytoplasm of the host cell. This genome of the coronavirus, which is also called RNA positive, is translated by the host cell machinery like ribosome into an antisense RNA. There are two things that could happen after this. One is that it can be replicated back into sense RNA, which is the same thing that came in with the virus. Or the antisense RNA can be transcribed through a process called discontinuous transcription. The antisense RNA can transcribe different mRNAs. The different mRNAs can be translated into different viral proteins, and this process happens in the rough endoplasmic reticulum. Once the proteins are made, they are sent with the RNA that was created in the first form of process into the Golgi apparatus, where they are put inside vesicles and packaged as a viral progeny. Finally, the new cell is released out of the host cell through exocytosis. Why is the coronavirus dangerous for our health? Although for most people, COVID-19 causes only mild illness, it can make some people very ill. More rarely, the disease can be fatal. Older people and those with pre-existing medical conditions appear to be more vulnerable. It is also very easily spread compared to many other viruses. Of course, it is also a new virus, so the human body is not immune to defending us from it. This is a graph created on January 21st. As you can see on the graph, when compared with SARS, even if the death toll was still lower than SARS, the number of cases was much higher, meaning that even though chances of fatality is lower than SARS, it is much easily spread, threatening more people. Our body's defenses against viral infections. The immune response to a viral infection is primarily generated by a type of white blood cell called lymphocytes. However, the number of lymphocytes that can recognize and react against any individual type of virus is initially very small. This is particularly true for a novel virus such as COVID-19, which people have never encountered before. In order to produce an effective immune response, the small number of lymphocytes that can recognize a virus must become more abundant. Even though lymphocytes proliferate quickly, it still takes several days before there are sufficient cells available to fight back against the infection. During this period, the virus may also be spreading rapidly, so there is a race between the virus and the immune system that may determine the final outcome in terms of recovery. In the early phase of a viral infection, a variety of immune defenses help to slow viral spread in order to buy time for the lymphocytes to proliferate. One of these defenses is mediated by a group of signaling proteins called interferons. When a cell becomes infected by a virus, the cell can recognize components of the virus and respond by releasing interferons that diffuse to neighboring cells and stimulate signaling pathways by binding to receptors on their surface. Consequently, these neighboring cells start synthesizing a large number of antiviral proteins that put the cells into a state of viral resistance. If a virus ever infects these cells, viral replication is inhibited. By this mechanism, the spread of infection through the tissue is slowed. Another early acting immune defense mechanism involves a type of white blood cells called natural killer cells, which are a group of lymphocytes that have the innate ability to recognize virally infected cells and cause them to die so that the virus can no longer replicate. Natural killer cells recognize alterations that occur in virus-infected cells, although they do not specifically recognize any particular virus infection. These innate immune defenses are critical in the first few days after infection while the lymphocytes divide and differentiate into cells that are able to mount an effective immune response. By 3 to 4 days after infection, there are enough virus-specific lymphocytes available to start mounting an effective immune response against the virus. In order to control the infection, the immune system must both destroy virus-infected cells and prevent the spread of virus between cells. These distinct actions are done by two different sets of lymphocytes. The antibodies that bind to the external spike protein are most important in limiting virus spread and conferring immunity. Cytotoxic T lymphocytes recognize and destroy infected cells of the body. Problems that scientists are facing in producing a vaccine. Despite the fact that it is a newly found virus, there are a few problems scientists are currently facing. One of them is the lack of very good animal models for testing. Following the SARS outbreak in 2003, scientists studied mice. Mice make antibodies in response to SARS infection and their immune system is well understood. But mice do not naturally develop pneumonia with SARS coronavirus infection, so it is impossible to test whether a vaccine protects against the disease. Hamsters develop lung changes on SARS infection but do not appear to get sick. Ferrets and several monkey species develop lung disease with SARS coronavirus but not consistently. These examples give some insight into the difficulties of producing vaccines. They show how important it is that vaccines stimulate the right immune responses and why safety tests are vital. Why did scientists quickly focus on sequencing the genome of the virus and shared their data? It all started when a virologist and evolutionary biologist from the US tweeted this first notice about the availability of what he referred to as an initial sequence of the virus. The virologist is a member of a consortium of the Shanghai Public Health Clinical Center that posted the sequence on an open access site. In a brief note, the website said researchers were free to analyze and share the data. After this, the analyzing began immediately. An evolutionary biologist at the University of Edinburgh calculated that the virus had a 89% similarity to SARS, as well as many other things emerged. The sequence of the virus is extremely important because you can see from the sequences how the virus spread, the speed at which it's spreading, and estimate the number of people that are infected. When the virus first emerged, people didn't even know what the virus was. Today, thanks to China's quick public release of the initial COVID-19 virus genome, there are now 18 genomes connected to COVID-19 that are being shared and studied by scientists around the world. By rapidly sharing this data, scientists were able to quickly identify COVID-19 as a novel coronavirus related to those previously found in bats. How to prevent something like this to happen again? There are three main things that we can do to prevent a pandemic like this in the future. Firstly, it is to invest in research and preparation. In 2018, the World Health Organization identified a list of diseases that it considered priorities for further research. Even when there are no pandemics, scientists can still conduct research beforehand to prevent pandemics. Secondly is to fund and continue to implement vaccine programs. While there may not be a vaccine for COVID-19 yet, large-scale vaccination programs are generally very effective at minimizing the rates of disease, while ensuring that health systems don't become overwhelmed with patients suffering from preventable diseases. Also, in the context of emerging epidemics, this is very helpful as it means focus can remain on understanding and containing new diseases. Finally, it is to strengthen health systems and show solidarity during outbreaks. We are only as strong as our weakest health system, and so ensuring that every health system around the world has the equipment and trained staff needed to provide quality care is crucial.